I'm Gina. I am a postdoc in Marvin's lab, and I am really excited to get to introduce Nancy Moran for today's talk. She's our VWR Distinguished Speaker for our biology seminar, and she is the Leslie Surgener Professor of Biology at UT Austin. Nancy is really an expert on symbiosis and host microbe interactions, and she got her bachelor's from UT Austin, then her Ph.D. from University of Michigan in zoology, and has a lot of recognition and awards for everything that she's done in her career. She's a National Academy of Sciences member. She's a member of the American Academy of Arts and Sciences. She got a MacArthur Fellowship, a Jim Teege Award from ISME, and lots of other things. So it's a real honor to have her here speaking with us today. And as someone, I did my PhD in an insect microbe symbiosis lab, and Nancy's work was something that I always really admired and have continued to admire in my career. She does such great work really kind of diving into the details of how microbes and insects and microbes and hosts are interacting, and that has a huge influence on how we understand our interactions with microbes. And she does a really beautiful job combining genomics and ecology and evolution and biotechnology and all these different fields to really fuel her work. And so thank you for coming, Nancy. And the last fun fact that I had was that she even has a mealybug endosymbiont named after her, the Moranella endobia. So I'm looking forward to your talk.
Well, thanks, Gina, for the nice introduction, and Marvin for hosting. It's been really fun so far, and I have to say this is the first talk I've given in 2 years. In fact, we were on our way here 2 years ago, and like in March 2020, and ended up not appearing. So here I am after 2 years, and the topics, I mean, I've added a little bit of new stuff that's happened within the last 2 years, but it's really just great to be able to talk to people in person again. It's really, you sort of remember why you like doing it. And so I've given a few Zoom talks, but this is the first one, like with actual people there. So I'm going to talk mostly about the gut communities in bees, social bees, especially the honeybee, the domesticated bee species that's around the world. And I've, as Gina said, I've worked on symbionts in insects for a long time, really from sort of the very beginning. A microbiologist called me up, Paul Baumann from UC Davis, and was all excited about the bacteria that live in aphids, and sort of the beginning of PCR and sequencing, and he just realized what these were, and I just dove into it and just sort of dropped whatever else I was doing and started working on symbiosis, and that was a long time ago. And I'm going to say a little bit about the other systems I've worked on, but then mostly talk about what happens in bee guts. So the main study systems in the lab, where we actually have the organisms, do lots of experiments with them, are these aphids and their symbionts called Buchnera. And that's the ones I first started working on originally with Paul Baumann. Paul Baumann named Buchnera. And then more recently, but still for more than a decade, we've been working on the gut bacteria and social bees. So I'll just say a little bit about these systems first, because they're very different. And in a way, they kind of frame my thinking, because they're a very extreme kind of symbiosis. They're very intimate associations. They're strictly maternally transmitted, and they're very ancient. So here's a picture. Here's an aphid sort of dissected open, and within the body cavity of the aphid, so they have this open circulatory system, there are these very giant cells called bacteriocytes. They're aphid cells that are filled with bacteria. And so this is looking inside one of those aphid cells. Here's the cell membrane of the bacteriocyte. And there's these large bacteria. They're about 3 microns spherical, called Buchnera. It turns out they're actually pretty closely related to E. coli, which was very exciting to Paul Baumann at the time, because it meant that there might be more clues that we could get from sequencing. And they're very important in aphid development. They're really part of the aphid. Here's an aphid embryo at an early stage. The blue shows the nuclei of the developing embryo. And basically the entire thing is filled. There's actually more symbiont than aphid in the embryo at many stages. So they're a dominant part of development. They're very influential. They're really part of the organism. And it turns out that this is kind of a syndrome. Here's just a little kind of cartoon of the situation. that these symbionts and hosts have co-diversified for very long periods of time. So aphids are about 200 million years old. They've been diversifying with them the whole time. Strictly maternally transmitted, so like mitochondria, really. They have extreme genome reduction. After this long periods of time, they lose most of their genome. So Buchnera has about 600 genes, and it's not even the extreme. They're maternally transmitted. And then there's a lot of evidence for what they do. They make limiting nutrients. So aphids feed on plant sap, which have very unbalanced amino acid profile from the point of view of animal nutrition. And the Buchnera make essential amino acids, so the same ones that we need in our diet, aphids need in their diet, and plant sap happens to be a very bad diet for that. So basically we know quite a bit about what they do, and this is all just from sequencing and different kinds of experiments, because one of the limitations of these systems is that we can't grow the bacteria outside of the aphid. These are, be like trying to grow mitochondria, which people used to try to do, but it doesn't work, and it certainly would be very challenging. And so this is all from different kinds of experiments. We find mutations sometimes in the Buchnera. We can introduce Buchnera between matrilines now, so that was sort of a breakthrough. So we can do some kinds of things and figure out this about it, and a lot of it's just genome sequencing. So it turns out aphids are just one example of hundreds of examples within insects of these kinds of symbioses. And so our lab and other labs have studied a lot of these. These are just examples, so cockroaches, some ants, somewhere in here there's a mealybug, I'm sure, but cicadas. So a lot of these tsetse flies have these maternally transmitted symbioses that have co-diversified with hosts, so people have done phylogenies. They have reduced genomes, and in general, they are supplying some limiting nutrients, either vitamins or amino acids. So it's a very common situation, and this is just a graph showing some bacterial genomes, so genome size in megabases on the bottom here, and then the number of protein-coding genes on the y-axis. So you see that in bacterial genomes, the bigger they are, the more genes they have. But it turns out that these endosymbionts are the extremes of tiny genomes. So all of the tiniest genomes basically are insect endosymbionts. Some other invertebrates also have these kinds of endosymbionts. So basically, Buchnera is not even the extreme. It's here. There's some that are quite a bit smaller. So we call this the symbiosis rabbit hole. Like, it's basically an irreversible evolutionary route where the symbiont lives in the host, is basically has no future elsewhere, loses more and more genes. The host is evolving probably to just prop it up, because the host has meanwhile become dependent on it, and so the host is evolving to keep this machine going. But it's in some cases probably things go extinct because they really are very degenerate genomes. The smallest one has 120 genes. So they still make their own ribosome, tRNA synthetases, and so on. And so that's most of their genes.
Are they all stuck to one?
Yeah, they're basically in one matriline, one insect. Sometimes you see more complicated. So sometimes they actually lose an ancestral symbiont and it gets replaced by a new one, which is interesting. So we can actually work out this kind of tapestry of symbionts coming in and out. Yeah. Oh, let's see. Let me see if I can. I'll try to talk louder too. I'll put it there. I'll see if I can talk louder then. Is that better? Okay, yeah. Thanks. Thanks for letting me know. Okay, so I'm not going to talk about those. So I'm going to talk about the gut microbes of bees for the rest of the talk. And it's something we've been working on since about, really, I'll tell you how it started with the first colony collapse in 2007, was the first time I even thought about bees. And so these are not maternally transmitted, but they are transmitted directly between individuals socially. So these are social bees. They live in colonies. They're encountering each other a lot, and they get their symbionts from each other. And these are gut symbionts. They're not living in the body cavity or within cells. They're within the gut. So they're not inside the animal, really. So it started back in 2006, 2007. That winter was the first time everyone started talking about bees dying. A lot of bees died that winter. It came to the attention of the world and the beekeepers. Never really figured out exactly what happened that year, but there's been ongoing problems with bee colony collapses. And this is the western honeybee is the main focus. So that's the domesticated species that has been spread worldwide by people, and pretty much everywhere people live, there are western honeybees. And so a few things happened that year. There was the honeybee genome was published. Colony collapse happened, and people were all worried what's happening to the bees. And also that was the very beginning of next-gen sequencing. So some early versions of it, 454 and so on. So it was the first time you could cheaply just sequence like crazy. And so a group of people got together and decided maybe there's a new pathogen that's hit the bees in the U.S., and we'll find out what it is by just sequencing lots of bees. And it resulted in this paper trying to look for a new pathogen in bees. And it turned out there was no new pathogen, and that isn't what happened that year. There's a lot of pathogens. They all seem to play a part, but why exactly that year there was an event isn't so clear. But the thing that came out of it, so this was just sequencing RNA from bees. These were potentially RNA viruses, so they used RNA. And so sequencing RNA, it turned out, and then later sequencing genomic DNA, that the main bacteria in a bee that were over 95%, and usually more than 99% of the bacteria in every bee, were the small set of taxa, about 8 species. And usually just 5 of them were in every bee. And so these different colors, these are just different bees, and it shows those same species. So each color, sometimes you see these diagrams, and each one is like, a phylum or something, or a class of bacteria. But these are actually very close clusters, that would be considered species-level clusters of bacteria, although that's a complicated thing, of course. And this was true of samples worldwide, so everywhere in the world, Europe, Asia, North America, it looked the same. So I thought that's really interesting. Here's a really conserved community. It seems to be sort of part of bee biology. The other thing about these bacteria is they weren't found outside of bees. All of the samples of these bacterial, based on 16S at that point, were from bees. So it suggested they were really host specialized and restricted, and every bee had them. So we did some more work, and just to summarize, so most of the bacteria are in the hindgut. So if you have the gut of a honeybee, insects have a foregut, midgut, and hindgut. These are in the hindgut, the distal part of the gut, and it's got 2 main sections called the ileum and the rectum. And there's this spatial organization, so a couple of main species are in the ileum, and then some others, including some gram-positives like Bifidobacterium and some Lactobacillus-type species, are in the rectum. And we mostly focused on this ileum section and on these 2 species, Snodgrassella alvi and Gulielmella apicola. And they live in a very tight biofilm on the surface of the gut wall in the ileum, with Snodgrassella sort of on the lower, like lining the epithelial cells of the gut, and then Gulielmella sort of in little clusters above it. And that's a repeated pattern in different bees, and you can sort of look at cross sections of the gut and see them arranged that way. So the thing that gave this life and that we could really get into it more is that it turned out we could actually culture all of these. And the trick was, and the reason no one had studied these before, is that they only grow under low oxygen or anaerobically. And so many people have worked on bees. The honeybee is a very well-studied organism. Behavior, metabolism, nutrition, it's important to people. But no one had actually ever studied these bacteria. And the reason is they were just culturing them in full atmospheric oxygen, and they would just it'd be the same thing as if you'd cultured human feces. You get really the rare things that were sort of weedy things there. And so a couple of really great people who were in our lab when we were at Yale University, Walden Kwong and Philip Engel, who both have their own labs now, were good microbiologists and got all of the species into culture. We gave them names, and so there's type species and all of that. So they're real bacteria that other people can and do work on now. We can sequence genomes of isolates, so we can get really known genomes, develop genetic tools, and do experiments with notobiotic hosts, so we can have known infections of known strains in a host and look at the effects. And this is just a picture. Another person who got involved more recently, a former grad student, Sean Leonard, developed the engineering tools, so adding different markers, and I'll talk a little bit more about what he did, but we can put in different markers and look at where they colonize in the gut and things like that. So I'll look at a couple of questions today. First, just some background. What do these actually do? Oh, sorry. He fell out of his chair in wonder. Okay. All right, Steve, yeah.
Sorry.
No problem. Be careful. Okay, so how do they affect the host, just in general? What are really the main reason people get interested in microbiomes in the gut is because they might affect the host, right? They're communities, they might be interested in how they assemble and so on. But really the main interest is what effects do they have on the host. That's why people generally study at least animal and plant-associated microbiomes. And then in the second part, I'll talk about how the more recent stuff about how they've evolved and diversified in hosts. Okay, so we've developed different experimental approaches. This is sort of a diagram just to illustrate kind of the things we can do. It turns out we can get germ-free hosts. So the newly emerged adult bees are basically germ-free. And so we can take them as pupae, let them emerge in a sterile environment, and then colonize them with known strains or with maybe the full microbiota that we get from the hindgut of another bee. And so we can have gnotobiotic hosts where we know what is in them, and we can do different experiments, see what happens. Or we can take colonized hosts that have a normal microbiota and then disrupt it and see what happens to them. And the advantage here is we can actually do experiments kind of in the natural environment. The great thing about honeybees compared to a lot of insects, you can put them in the hive and they will stay there, or they'll at least keep coming back there because that's their home. And so we can do these marker recapture studies over time in a natural environment to see what are the effects on the host in the hive as opposed to in the lab. And so we can only do this with non-engineered strains, but still it's very helpful. We can look at what happens. So I'll just give you some examples of how we've done these things. So this is just some pictures of people, students in the lab developing the gnotobiotic host. So basically these are normally developed in the hive. They pupate in the hive. We take the frame, the honeybee frame in and pull the pupae out before they've started to emerge on their own, and then just let them emerge in the lab. And when they come out, they're basically completely germ-free. And this is just an illustration of a naturally exposed in the hive after 4 days. And the purple here is one of the key species, Snodgrassella, that lines the gut. So it has these kind of these invaginations along the length of the ileum. So there's these 6 lobes, and you can see the Snodgrassella sort of lining the lobes of the gut. And here's one that was just mono-inoculated with one strain of Snodgrassella after 4 days, and you get this really robust colonization. So we can basically colonize hosts in the lab and get really robust colonization. And doing that, we can show different things like what they do in the bees. So one thing, and I think is probably the most important role of these gut bacteria, is they defend against all these opportunistic pathogens. And I'll just talk about bacterial pathogens. So Serratia marcescens, common insect pathogen, is in every beehive has some level of it, but it's usually just very low and is not causing pathogenesis. But if there's a perturbed gut microbiota or no gut microbiota in the bee, it will be invaded by these and it will have a higher mortality. And it seems to be through direct suppression in part. So here's a graph where bees with different combinations of bacteria, so microbiota free, they have a very high level of Serratia in the gut. And then with single strains of the gut microbiota, even single strains one by one have some inhibitory effect, but all of them together have more of an effect. And this is the full gut homogenate, the full diversity, and it has a more protective effect. And then over here, this is if you start with if you compare conventional and microbiota free, again, you get this very big difference. If you perturb it with tetracycline, you get a reduction in the protection of the conventional one. So basically, this is one example. This is from Margaret Steele, a former grad student that I shared with Marvin at University of Texas. She's now at WashU doing a postdoc. And she and others have done these kind of experiments repeatedly. We get these protective effects. And people have also shown effects for protection against RNA viruses. There's a microsporidium that's important in bees. There's a trypanosomatid. And in general, having a robust normal microbiota protects against this range of pathogens. And it's probably through different mechanisms, some involving the host immune system, triggering the host immune system, others direct competition or just physical barriers to where they colonize. So it's probably a range of mechanisms because these are really quite diverse organisms. So one thing about the engineering, we can actually visualize in the gut, where the colonization occurs. For example, here's this pathogenic Serratia. In a microbiota-free bee, it's just everywhere. It just replicates like crazy. But when Serratia is present, when Snodgrassella is present, it lowers the titer of the Serratia, and it seems to actually just block its colonization along the gut. So another kind of experiment are these mark recapture. So we can take bees from their hive, and they have a normal microbiota, and then we can have control bees and experimental bees that we give, say, an antibiotic, a disrupting chemical that will disrupt the microbiota, mark the bees based on control or antibiotic, return to the hive, and then recapture them over time, so up to a week or even longer, find them over time and see who. It's pretty much nature from the point of view of a honeybee living in the hive. We can see what is their fate out there, and so we can compare it for host survival. We can look at microbiome composition over time, and then pathogen susceptibility. We can do lab trials. And first person who led some work on this was Casey Raymond, who's now at University of North Carolina, Greensboro. But she used tetracycline and showed that when you do this mark recapture, the ones with the perturbed microbiota have much lower survivorship over time, and they have greater mortality. If you do a lab trial, the tetracycline ones have a greater mortality when exposed to Serratia compared to ones that don't have the tetracycline and Serratia is this bottom line down here. So it makes them more susceptible. And then another former grad student who's now a postdoc in the lab, Eric Mota, did similar stuff with another chemical, glyphosate, which is an herbicide widely used, like formulations like Roundup and others use this, and it's the target of glyphosate is an enzyme in the shikimate pathway for aromatic amino acid biosynthesis, which animals don't have. We have to get our aromatic amino acids in our diet, so that we don't have the direct target of this chemical, and nor do bees. But most bacteria, plants have it. That's why it's a herbicide, but also most bacteria and fungi also have it, and so it has this potential negative effect on all these other organisms. And it turns out that Snodgrassella in particular is very sensitive. It has a very sensitive enzyme that is the target of this, and so when glyphosate is, when bees are exposed to glyphosate, there's a big effect on the microbiome composition. This is just a PCA plot showing the community composition is represented by the position here. The glyphosate-treated ones diverge from the non-treated ones. And Snodgrassella in particular is really reduced, almost eliminated by it. And it's really a key species in the bee gut. And if you do lab trials for assays for pathogen susceptibility, it makes the bees more susceptible. So it's just another way that this can be disrupted, and it shows this importance, and also raises the question. It's all a question of concentrations, of course, and Eric has done trials that do resemble concentrations found in real environments, but we don't know really outdoors how much of a factor this is. So other effects. There's other effects on the bee development. And so when the bee emerges as an adult honeybee, it gains weight afterwards. It eats a lot and it gains weight. And that's just the normal part of their biology that's well documented. And bees have this sucrose response, whereas if they encounter a liquid that has a certain amount of sugar in it, they will respond by putting their proboscis out. And this is a standard sort of behavioral assay on bees. And so you can put on their antenna a given concentration of sugar, sucrose solution, and see if the tongue goes out, and that's the proboscis extension response. And it turns out that the ones that are microbiota free are much more eager to feed, and they will respond to lower amounts of sugar, whereas the germ-free ones or microbiota-free ones have a very hard time responding at all, and so they don't eat much, and therefore they don't gain weight as much, and they have a very different physiology. It also affects their insulin signaling, looking at gene expression of insulin genes. And it has a huge effect on the short-chain fatty acids that are present in the gut and hemolymph. So you basically just totally changes the composition, both in the gut compartments, the ileum and rectum, but also in the hemolymph, the body cavity. So, like in particular, butyrate is absent in the microbiota-free ones and is the dominant short-chain fatty acid in the hemolymph when they have a normal microbiota. So it has a very big effect on the bee physiology. That was work by Hao Zheng, a former postdoc. So, of course, genomes tell us a lot about what they may or may not be doing, and it turns out if you look at Gilliamella and Snodgrassella, they have very different metabolism. Gilliamella is a fermenter, it uses sugars, whereas Snodgrassella just uses short-chain fatty acids. And so there's this potential for kind of, not mine, is it? No, okay. There's this potential for cross-feeding and sort of a complementarity between them. Snodgrassella is an obligate aerobe. It has to have oxygen, low amounts. It doesn't grow in full oxygen, but it needs some, whereas Gilliamella is a facultative anaerobe. And so we got ideas we've sequenced lots of genomes at this point, and they give you some ideas about who's doing what. But just looking at the genomes doesn't really tell you exactly who's doing what. Although we can say for sure that Snodgrassella cannot use sugars. And so this is kind of an illustration of what we generally think is happening, mainly work from Howe, because he did a lot of metabolomics in the gut. That basically you have your host epithelium. There's an oxic zone there from the host cell, so there's some oxygen. But Snodgrassella's metabolism is basically sucking up the oxygen, creating this anaerobic zone. So the germ-free bees don't have an anaerobic zone, whereas the normal microbiota, they do. And then it's leading to all this fermentation. And so the pollen cell walls there's actually material that has gone through the midgut of the bee that still is digestible by bacteria, but not by the host, right? So different enzymes are needed for breaking down these complex polysaccharides that are especially in the husk of the pollen. I forgot to mention, bees only eat nectar and pollen. That's their diet. So nectar, they get sugar. The protein's supposed to come from the pollen, but the pollen husk also has polysaccharides in it that is a potential source of energy, and it seems to at least be providing some energy because some of the short-chain fatty acids end up in the hemolymph of the bee. So we don't know how much of the nutrition this is, how important it is in nutrition, but it's at least something. They also have a big effect on the pH. The pH is very different with and without the microbiota. So on. This is just an image of the cross-section of the ileum. So again, you have these lobes out, and you can see the Snodgrassella is sort of the light color there. It's lining the gut. The Gilliamella, depending on the strain, they'll occur in these little clusters of like 50 cells, so little clusters that are on particles of polysaccharide stuff from the pollen husks. So here's a pollen husk that's intact that comes through the midgut. So there's a lot of undigested stuff. It's a lot like humans. There's a lot of undigested sort of plant fiber that ends up in the distal part of the gut that bacteria can work on, and that does happen in honeybees. So Gilliamella strains, they have, depending on the strain, which I'll get into, they have a lot of different enzymes for breaking down polysaccharides. For example, breaking down pectin, which is, so there's this polygalactoric, the backbone, PGA backbone of the, so there's an enzyme for that, the pectate lyase, and there's all these enzymes for breaking the side chains and the sugars off, and then there's a bunch of genes for transporting different sugars in and using particular sugars. So there's this huge sort of set of genes that can be present in Gilliamella strains. And in general, Gilliamella apicola always has the ability to break the backbone and at least digest pectin in part. So these are secreted enzymes breaking down this extracellular polysaccharide. So if we look at Gilliamella strains in particular, like many bacteria, there's this large accessory genome, right? There's a pangenome that includes a large set of genes, whereas any one genome doesn't have all of those. And so this is looking at a bunch of genomes. If you look at the shared genes, it decreases over time, and so there's about 1,500 shared genes, but there's all these extra genes that can be in particular strains. And it turns out a lot of those extra genes are involved in using polysaccharides. Some of them are involved in toxins, antitoxins, sort of microbial combat, but a lot of them are involved in dealing with the environment that is imposed by the host diet, this different stuff that comes in. And so given strains, some can, they can use different polysaccharides, some can use hemicellulose or pectin and so on, different sugars. Yeah. These are all from the western honeybee, but in general from different individual bees, although they could be in the same bee. These are just the genomes that were available there as isolates when this was done. We now have quite a bit more, but it's you're pretty much saturating up to about 4,500 total for the pan genome. So this gets to the second question: how do these host-restricted symbionts evolve? and specifically, how do they diversify in their host? So we started getting interested in these questions of how the strains differ, how much diversification can happen between and within hosts, how host restricted are they, and so on. And we knew something about this early on, because even at the very beginning, when we first realized that the Western honeybee has this set of conserved lineages in the gut, we got samples also of bumblebees, which are related to honeybees, but rather distantly related. So this is the genus Bombus. It's a different genus, but it's in the same clade of bees, and they're also social, and all their ancestors have been social. And so we sampled a few bumblebees, and they had some of the same characters in the gut, including Snodgrassella and Gilliamella. So every normal bumblebee also has a Snodgrassella and Gilliamella. And if you look at a cross-section of the gut, it's got very much the same arrangement in the gut. So it seems to be an ancestral situation that has evolved in these different lineages over a long period of time. And Walden Kwong, who was my grad student, he just took a job at the Gulbenkian Institute in Portugal. He's got his own lab there. He got interested in the evolution of this, and he got a lot of samples, which involved some traveling. He actually got dengue in Asia looking for bees, and you know, it was quite an adventure. But he got a bunch of bees and worked with entomologists in different parts of the world, and looked at basically lots of phylogenies. And this is just a summary. So he did phylogeny. He actually grew isolates. He took these back in glycerol, grew them up, and this is sort of a summary of what his findings point to. Is that at the beginning of this group of bees, social bees, which includes honeybees, bumblebees, and another big tropical group, the stingless bees, this is where sociality originated. These are all social. They have a queen and the workers. And it's also where a bunch of the core microbiota colonized. So 5 of the species seem to have colonized back then and evolved with these bees over time. And lots of things are consistent with that. The amount of divergence between the strains, the phylogenies of more or less map onto the bee phylogeny, although not perfectly, but more or less. So it really seems that there's been this long-term evolution of this whole set of lineages with this set of bees. And then sometimes additional ones have moved in. So in Western honeybee, here's an extra one, here's an extra one that have moved in that are restricted only to a smaller set of hosts. So there's this long-term evolution, and it seems to have resulted in close coevolution between the host and the symbionts. And one indication of that is if in the lab we can use bumblebees and honeybees as recipients, so you can grow bumblebees in the lab too, and we can see if they can exchange symbionts with each other. And basically, you cannot colonize a bumblebee with a honeybee strain or a honeybee with a bumblebee strain. So they're host restricted. We call them both Snodgrassella, but clearly they're different. They should be different species, and they are not able to colonize each other's host. And there's lots of opportunity for that to happen in nature. They go to the same flowers, they meet all the time, but it doesn't happen. It seems that there is some ability for them to colonize like closely related honeybee strains. So in Eastern Asia, there are other members of the Apis genus, and some of those can colonize the Western honeybee. Same with Bombus. There are some you can go between species sometimes. So it's not an absolute barrier as soon as they've diverged, but it seems to be largely a barrier. So recently we got into trying to look into how diversification has happened in these Snodgrassella and Gilliamella in particular over this period of time, and how have they diversified both within and between hosts. And a lot of this work is from he goes by Y.Y. Lee, who was a former postdoc. And basically it turns out, so here's a phylogeny with a lot of the isolate genomes that we have available, 130 isolate genomes, some of them from bumblebees, some of them from honeybees, some of them from the Asian honeybees, all this yellow is from the Western honeybee, so it's by far the best sampled host species on this. And it turns out that you can look at this and you can infer that there has been diversification sort of at the species level, both within hosts, within a single host lineage, and also between hosts. When the hosts speciate, the bacteria also diverge. And this is kind of just a little subset of that to make it a little more obvious. Another group in Norway described a new species of Gilliamella, recognizing that within every single Western honeybee, there wasn't just one Gillumella species as we first sort of, just to simplify things, we kept them as one. There were actually two. So we had Gillumella apicola as the first one we described. Walden Kwong led it. But they also, Jane Ludvigsen also described this additional species that was actually already in our data as well, but recognizing that it was distinct, Gillumella apis. Well, why call it a species? I mean, I think we'd actually end up naming a lot of species if we really got into it here. But these are interesting because they're really quite distinct. And also you can see that they're both present in both the Asian species and in Apis mellifera, the western honeybee. So this divergence, this original divergence probably happened within a host, and then as the hosts diverged, they diverged with them. And so there's both, sort of both of these things going on. It seems that the Asian species and the western honeybee, they both have Apis and apicola, both species. Sort of interesting. And then we noticed some other differences about them. Their optimal pH requirement was different, and they also differed in whether or not they could break down pectin. So apicola can and Apis can't. So it looked like there really is a different ecology for these two that have diverged within the host. So this is a diagram looking at the ANI, the average nucleotide identity, and it's looking at apicola here and Apis here. And if you just look in general, you see that within each of them, there's high identity, so it's generally above 90%, often above 95%, so they're really quite close in terms of, these are looking at shared genes, how identical are the sequences, whereas between them it's quite a bit lower, and that would go along with kind of your usual picture of these are different species between them, right? Even within them, there's probably multiple species, so if these population designations here come from an analysis to look at amount of recombination, and it looks like there's sort of non-recombining sets even within these, but certainly between them, they're quite different, and they seem to be different, and there's likely more species involved as well. Initially, they were about 97% identical for 16S, which is what we first look at, and that suggests different species. That's pretty divergent, right? And so, but we just didn't want to bother with too many names at the beginning, so we just designated the one species. But it is correct, I think, that at least two should be recognized. And so this is based on an analysis, this pop cogent analysis, which I don't want to go into, there's not enough time, but it basically looks at whether or not there's, if you take homologous genes, the homologous part of a genome between different strains, is there evidence for recombination between them? So if you have variants at different locations, are you sometimes getting recombination that puts those into the same genome? And it's just one approach to doing this that tries to designate recombining populations, because these are bacteria, they're mainly clonal, but there is homologous recombination as in most bacteria. And you come up with these sort of clusters that can include recombining strains. So here's a big cluster within Gillumella apis. Here's a big cluster, population 1, that includes all these strains. And I'll note some of them are from Arizona, some are from Norway, some are from different places, so even Asia. So this cluster is not defined by geography. It's basically just a recombining cluster within this one species. And then these may or may not be other. There's not enough sampling to really say. But so let's call this a species. They're recombining, and they're really quite close to each other in terms of sequence divergence. And then apicola is another species, and maybe a couple of species. And again, these are occurring. There probably are two species here, and they're occurring in Europe, North America, all over. And these are also Europe, North America. So it looks like there's these clear clusters, but there's definitely no recombination between apis and apicola if you look for recombination. So there's no homologous recombination as distinct from lateral gene transfer, right? That happens between everything, but this is homologous recombination. So there's zero combination between them. Another point is every honeybee has both apis and apicola in it, so they're co-residing in every bee. And they also co-reside within this other apis species, Serrana, which is the most widespread one in China and other parts of Asia. So if we sequence genomes, we can see there's metabolic differences. One of them is that apicola, but not apis, has all these genes for extracellular pectin degradation that I mentioned before. So basically, can break down all these different side chains and the main backbone, and it has transporters for the different sugars. And so basically, it has ability to deal with these polysaccharides that Apis lacks. And Philip Engel, he early did studies actually confirming that when they have these genes, they do break down pectin and they do use different sugars and so on. So basically, these are very good predictors of what they're actually able to do. How Zheng did some of that too. And then in the other direction, Gillumella apis, but not apicola, can use urea nitrogen. So basically, it has these extra genomic islands that allow it to take up urea and utilize it. And so that's one thing it can do that apicola can't do. So they have these differences in their capabilities. And that might have to do with we thought potentially that has to do with different parts of the gut where different things are going on. And from Hao Zheng and study, we knew that the pH changes as you go through the gut, the amount of all kinds of things change. And one thing that's really important in insects that changes is if you look at the gut, so here's the midgut, then here's the ileum. So right here at this pylorus region, that is where the Malpighian tubules, I don't know how many people know what a Malpighian tubule is. I have to teach it to my own. Anyways, the insect excretory system is how they get rid of waste, right? So they don't have, they get rid of waste through the digestive tract. They dump it in at the beginning of the midgut right here. And so this is basically nitrogen waste in the form of uric acid and urea. And so being able to use urea might be especially important for something that's living up in this region. And so that might be apis, whereas digesting the polysaccharides and stuff is something that's back in the back part of the ileum and in the rectum. And so that might tell us that perhaps apis and apicola are living in different parts. And so we looked into that using some fluorescently labeled strains that Sean Leonard made. And it turns out that apis, so here's the ileum, here's the pylorus region, and here's the back end. And so apis is really totally restricted to this pylorus region. It doesn't occur back here at all. And then here in pink is Snodgrassella is in pink, and this is apicola. So apicola is a little bit all along it, but more at the back end. And then here's one where they're all three are labeled together. So this was colonized alone. This one is with these two, and this one's colonized with these three. They're different guts. And this, so here's apis up at the pylorus, and Snodgrassella all along the whole thing, and apicola back here. So these are like intact guts that are not sectioned or anything. They're just, because they're fluorescent strains, you can still visualize it. So they're really in very different parts and really not spatially close to each other. And if you wonder, like, what will give rise to a barrier to homologous recombination, you can't just have different ecology, because if you're living side by side, the DNA is still going to go back and forth, right? You need to be apart from each other, and it looks like they are generally regularly apart from each other. Also, their pH preferences also correspond to these differences, so it looks like a real difference. So these were with lab ones artificially colonized with just a few strains. But even in hive bees, if we look for the urease genes, which are a marker for apis, we find that they dominate in the pylorus. That basically, even in just wild bees taken from the hive, you still get this pattern. So let's see where we are here. So this is kind of a diagram, a cartoon of what maybe is happening with Gillumella. Basically, there's some divergence within the host, and this happens say, for example, apis gets the ability to use urea, apicola gets the ability to use pectin or other polysaccharides, and they start to live in different parts of the gut as a result and be spatially separated, and that leads to just basically bacterial speciation. And one thing I didn't emphasize, but in the urease genes were acquired by horizontal transfer. If you do phylogenies, they came from some other bacteria, and these pectate lyase and ability to use these polysaccharides also come in through horizontal transfer. So that's playing a role in enabling them to sort of acquire and develop these different niches. And then in apicola, maybe we're getting some splits again. And so basically you have divergence within a host, but then each of them also diverges when their hosts diverge, right? So if between, say, this apis species Serrana and mellifera, they seem to be distinct strains, and of course they're isolated because they're in different hosts. Snodgrassella is a little bit of a different picture. They haven't diverged and formed all these niches. They seem to just really be one strain within a host, essentially. And the main differentiation we get when we look at these recombination analyses is geographic. So in Europe, there's this one cluster, and so that may just be that, yeah, we've sampled in different places and they haven't met up lately, so they haven't recombined. It may not represent any kind of speciation at all. And they're very close. If you look at the average nucleotide identity, they're all very, very closely related. So it seems like it hasn't undergone these splits, and that it's really just within a single host species, at least, remaining as a single bacterial species. And it's one, Snodgrassella is much more intimate with the host. It's really, its main environment is really defined by the host. It's right on the host epithelial wall, so perhaps it's there's fewer sort of ecological niches that are based on the environment and the diet. Yeah, so in Gilliamella, that it doesn't directly adhere to host tissues. It's mainly really influenced by what's coming in the diet. There's this spatial heterogeneity, and it's got horizontal transfer, so it's diversifying in this one way, and Snodgrassella is much more just following along with the host lineage. So it brings a question, though. We did show that in Snodgrassella, they can only colonize, they're very host restricted. They can't jump between hosts very readily. So they have an intimate interaction with the host that's preventing them from jumping between hosts for the most part. And we don't know what that is, but it does do some funny things that I think indicate some kind of really close interactions with the host that we don't know what they are yet. And this is one case of it. Richard Horak, who was an undergrad in the lab, he did his honors thesis, but one thing that these bacteria do is they stimulate the immune system of the host. I haven't talked about that a lot, but if you have a gram-negative cell wall with peptidoglycan, it's going to stimulate the insect immune system, and the toll pathway and the IMD pathway and all of that come into play. And so one issue about the host accepting a symbiont is, does the immune system exclude it or not? And it's kind of interesting because if you look at heat-killed Snodgrassella or heat-killed E. coli and you give them to a bee, you stimulate, so that would be the blue and the green bars here, you stimulate these genes that are in the IMD pathway, right? But weirdly, when you give them live ones, it actually somehow suppresses that activation. So Snodgrassella, when it's alive, is able to tell the host to calm down and be less reactive. And we don't know how it does that. This is something he did during the pandemic, and we haven't figured it out yet. But somehow it's probably got some really intimate interaction with the host that is allowing this. And it may be we think potentially it's actually delivering molecules to the host, even RNA and things like that, that could be suppressing it, but we don't know at this point. One reason we think that, I'm going to just end with this little thing. We've done a lot of work engineering the gut microbes to try to influence the bee, and this is mainly work of Sean Leonard, now joined by P.J. Larib, who's replaced him basically because Sean's moved on. And so basically we can put different constructs into Snodgrassella in particular and have them expressed in the host. And one thing we're really getting into is double-stranded RNA that activates the RNA interference pathway of the host. And Sean did some work on this, and it showed that you can reduce viral loads of RNA viruses by targeting that virus with a specific sequence, activating RNAi and lowering the viral load. It actually can increase survivorship of the bees, but you can also knock down the bee genes, and we sort of got interested in that as a genetic tool in the bees. So Snodgrassella in particular is able to do this. And so maybe it has some particular ways of influencing the host, and we're just kind of tapping into those, even though we don't know how they work, of delivering things to the host that then influence its biology. And so it's likely there's some complicated biology there that we don't fully know about. This is just showing some of the results from knocking down the bee virus. We can basically give them these symbionts that have expressed this double-stranded RNA that targets the virus, and we can knock down the viral loads and make the bees survive better. This was work that Sean did earlier. So I'm going to just go to the conclusions. So just to summarize, I don't want to go over that this bee gut system, I've just kind of given you some highlights of the different things that can be done with it. It's a simple, defined gut community that's useful for studying different aspects of interactions. It's got major roles in bee biology, which I think makes it of more interest to more people. It's a defense against pathogens for sure. If you perturb it, they're more susceptible to dying. I actually think that might have been what happened in the colony collapse in 2006, because a new antibiotic was introduced to control there is a bee pathogen that's a bacterium. And a new antibiotic called tylosin was introduced that year. And previously, they'd only used tetracycline. It turns out the bee gut microbiota in the U.S. has a ton of tetracycline resistance because there's a long history of tetracycline by beekeepers. But a new one was introduced 2006, and that's when all the bees died. And it turned out they died from all kinds of different pathogens. And it could just be that they were all disrupted and therefore more susceptible to whatever was around, and then had a greater mortality that year that was notable. But they also play a role in nutrition and metabolism. They're diversified with hosts for 80 million years through this social transmission. And then speciation seems to be this complex thing where they can actually acquire different niches within a host. And then, of course, they also will diverge when the hosts diverge as well. And that probably really differs. So Snodgrass and Gillumella, they seem to have different modes of diversifying. So just to acknowledge, here's a bunch of people I tried to mention, most of them as I went along. Toby Hammer, also, he's working on the gut microbiomes of Bumble Bees. I didn't talk about it as much, but so all of these are now have their own labs and working on bee gut microbiomes, so I can let them do all the work in the future. And then Eric Mota, I also talked about him. He's still in the lab, and Sean has moved on to Lawrence Livermore National Lab, and Eli is a technician in the lab who's worked on all of this, really very central to all of this work. And with that, thank you.
If people have questions, we can pass around the mic so they get picked up and Nancy doesn't have to repeat them.
Right, thank you. Is this on? I can't tell. Anyway, great talk. I had a question about solitary versus social, in the sense that you show that you have this deep homology to this gut taxa that seems to go back to the origin of eusociality in the Hymenoptera. But of course, the vast majority of bees are still solitary. And I'm wondering, given that they don't have the opportunity to maintain a colony through winter, they often just provision some larvae, in a stick or a hole in the ground, and then peace out for 9 months. Do they have totally different gut microbes, or are these things able to be deposited on the inside? And
Yeah, it's mostly totally different. Yeah, so really early on, when we found this in honeybee, we did get some other bees, and we found that bumblebees had kind of the same things, but other bees didn't at all. And they will tend to have more like lactobacillus that are also present in the environment, that also grow outside of the bee, and some acetobacter and lactobacillus. There's some exceptions. So Carpenter bees, which are really cool bees, but they're solitary, although they do nest in kind of aggregations. It looks like they also have a distinctive microbiota, but most solitary bees don't, and they mostly have a smaller microbiota, so I didn't emphasize it, but there's quite a lot of this microbiome in a honeybee. It's a large part of the bee, much larger. And then if you go out to like wasps and stuff, which are like bees were wasps before they became bees, they're carnivorous. They tend to have very little microbiota, and kind of erratic, just more environmental microbes.
Hey, so going back a little ways, you mentioned the work where you showed that glyphosate was messing with one of the species and making the community as a whole more susceptible to pathogen invasion. Do you have any idea what it's doing with that one species that makes that species so vulnerable? If I recall correctly, that has to do with like amino acid synthesis.
So the target of glyphosate is one enzyme in the shikimate pathway that goes to tryptophan and phenylalanine and things. And actually, it turns out that it's a little more complicated because there can be sensitive or resistant versions of glyphosate, of that enzyme. Sorry, and that's why you have like the Roundup Ready plants. You put a resistant enzyme in them, and then they're not. And so. And initially that was done by picking bacteria that had a resistant enzyme. But it turns out that these bacteria do vary Snodgrassella has a sensitive one, so do most of the, and for some reason Snodgrassella is impacted more than some of the others. They're in a biofilm, everything's, it's hard to say exactly why, but it's been very, I showed just the results from the first study that Eric did, but he did a bunch of other studies looking at dose response and lots of different things with concentrations because we felt like we should follow it up. It's very consistent that Snodgrass ella gets knocked way down. And so he took the enzymes out of these and put them in E. coli and looked at the sensitivity of the different versions of this enzyme and could verify, that they were. So, yeah.
Thank you for a really great talk. So I'm really interested in how these microbes could be influencing resource use and sort of the breadth of resources and nectar resources that bees could be using, maybe ancestrally in wild populations, and then how we've changed sort of selection regimes now. And, we cart these bees to particular nectar resources, and in nature, that figure you showed with the tongue response actually responsive to a broader breadth of sucrose concentrations with microbe was so interesting. So I wondered, do you see any signatures of microbial change that you think might be linked to how we're coercing bees into a narrower
I think the main
diet?
one is that in beekeeping, a lot of bees are thrown together all the time, whereas in nature that would not be. Usually bee colonies reproduce by basically fission. The workers, they make 2 colonies out of one and they make a new queen to go off. But in beekeeping, all kinds of queens and workers are thrown together all the time. And so there's I guess one thing I've wondered about, it's not so much resource use, although it could affect it, because just more opportunity for horizontal movement of some of these genes that are accessory genes, but also just the selfishness of the microbiome. I mean, they're less tied to the future of that particular colony, right? Basically, they're competing all the time in new combinations and being thrown together. And so there could be sort of more kind of like selfish evolution of the microbiome, and that basically they're replicating as much as they can and causing diarrhea in their host just to get into the other bees. It's a fecal oral route. So how do you get into other bees? So it's possible that some aspects of beekeeping sort of encourage negative evolution of these strains, because basically they're trying to spread in competition with other strains that have been thrown in with them. But that's the main thing I've thought about, yeah. But.
Thank you.
I was wondering, you talk a lot about the role of the microbiome in metabolism, but I was wondering about detoxification, because pollen and nectar can have a lot of funky chemicals in it, because plants don't necessarily want every single insect to be able to eat it. And so, because I remember reading a paper, I think it was solitary bees, where they fed them a bunch of different pollens, and they could only be raised on certain ones. And so I wonder if these really robust microbiomes that social bees have is also helping them to be more generalist compared to solitary bees.
Possibly, yeah. We have done a few experiments, and some other people have on like ability for the microbiome to break down toxins. It's not super clear cut, but there is some. Like there's this one compound, amygdalin, which is highly toxic when it's degraded through certain routes, and through the microbiome could potentially make it worse, because actually there's some routes of degradation that make it more toxic, some that make detox, and it does definitely have an impact, and potentially having a diverse and the genes that are doing this tend to be accessory genes, so erratically distributed in strains, not characteristic of a whole species. So potentially having all these strains is protective in that sense, that's part of a role. We did stuff like, can they break down like some of the widely used insecticides, and we didn't find positive results. They can't. So things, so it's possible they are. Some sugars are actually toxic to bees, like arabinose and stuff are toxic on their own, and it does look like these bacteria can metabolize those and take them up and perhaps protect the bee. One aspect is these are in the hindgut, so the bee is mostly absorbing things in the midgut, so that may limit the ability of the microbiome to save the bee from toxins if they're already absorbed, but it might have some effect, yeah.
Hi, Nancy. You mentioned 80 million years ago there was the emergence of this microbiome and coincidentally the eusociality of bees. Do you think there's a link between those two things, or is it just coincidence?
Yeah, well, I think the sociality enabled the specialization of the microbiome, because they don't replicate outside the host, so how do they get from host to host? And most insects, as mentioned, solitary bees, they put a wad of pollen and lay an egg on it, and then they leave, so it's very hard to transfer the bacteria. And also they also didn't emphasize, these are only in the adult bee, they're not in the larvae. So how do you get from one adult to another in a really reliable way? Well, in honeybee, it's easy. As soon as they come out, they're interacting a lot. And that's true in all these social bees. Whereas in solitary bees, they're pretty much living life on their own except for mating, so they don't have these interactions. So I think probably sociality enabled this. And it's, termites also have them, very similar. They transfer them within colonies, and they don't necessarily need to be eusocial, like with the queen and the workers being sterile and all that, but they need to be regularly, meeting up right away after they emerge. Potentially, I mean, it would definitely make it beneficial to be around, your relatives or other members of your species right away. And so potentially, yeah. I think that was one of the really early ideas for the evolution of eusociality buried somewhere for termites. Yeah.
Hi. I was kind of wanting to go back to that previous point you made in terms of like kind of maybe like almost like a selfish evolution of the microbiome. How does this symbiont get from one host to the other in the most proficient way? And that got me thinking, is there any like scenario, any kind of parameters in which this symbiont will flip to the other side of the spectrum and behave more, perhaps parasitically, or, I'm kind of thinking on it in terms of like the parasite mutualism spectrum. Like, is there any, or have they like kind of co-diverged like long enough to where that's not really an issue?
I mean, I think there's always the potential because, there's many mutations that would lead to sort of more replication than is ideal for the host and so on. So, and some of the Gilliamella strains are actually associated. I mean, it seems like sometimes Gilliamella, for example, I think over-replicate. I mean, it seems to be a burden on the bee. There's at least some indication of that. And, so it's possible that they're always kind of, just walking this tightrope because, I mean, obviously if you kill the bee right away, it's not good, but for the symbiont, that is. Basically being transferred, I mean, the highest level of transmission may somewhat correlate with over-replicating that is not good for the host. So.
Thank Nancy again one last time for a really interesting talk, and thank you all for coming.